Now that we've taken a look at the process of gene transcription and how messenger RNA transcripts are modified by alternate splicing in eukaryotes, it's now time to take a look at gene translation, so the molecular details of this process. Okay, so as with transcription, we're going to take a look at the molecular players. Then we'll examine the process of polypeptide synthesis in detail. And then we'll talk a bit about completing proteins and how proteins get targeted to different regions of the cell. Okay, so first our molecular players. One of the big molecules involved in the process of translation is a type of RNA molecule that's referred to as tRNA. And the T in tRNA stands for transfer RNA. So these tRNA molecules are shaped as shown in the depictions down here on the slide. We don't need to worry too much about a lot of the details. There's some kind of internal structure to these where the RNA actually forms hydrogen bonds with itself. What really matters with these tRNAs is there's these loop twisted regions, but then there's one particular region that's highlighted here, which is referred to as an anticodon. So this is a region that's going to be complementary to a codon on the messenger RNA. And then along with that, the other major business component to this tRNA is a location where an amino acid can attach to this tRNA. And next to this cartoon depiction, there's also a three-dimensional depiction of what these tRNAs look like. And there's other depictions that can be used as well. So then you might wonder, so how do the amino acids actually get attached to these tRNAs? And you don't need to remember too many of the details of this process at the moment, but just to satisfy your curiosity, the amino acids are actually attached by a type of enzyme that's called an aminoacyl tRNA synthetase. So synthetase means to build, and aminoacyl tRNA refers to the process of attaching the amino acid and forming an acyl bond on the tRNA. So basically, these get abbreviated AARS, and these will attach a fresh amino acid onto a tRNA. And each AARS is specific to a specific amino acid. So there's kind of a lot going on behind the scenes here. Along with that, the real workhorse molecule for the process of translation are ribosomes. So ribosomes are also composed of a combination of amino acids and RNA. They're a fairly large globular molecule, and they're actually put together in the nucleus. And what ribosomes do is they are an enzyme that brings together tRNA with the mRNA. So their job is to help with matching up the anticodon on a tRNA with the codon that's present on an mRNA. And so again, they're built from a combination of RNA and protein. So the RNA found in ribosomes is called rRNA for ribosomal RNA. And they're actually constructed from two different subunits, so distinguished as being the larger one and the smaller one. So big subunit, small subunit. And the cartoon that's on this slide also, so it shows you kind of the blobby structure of the ribosome. And then it also shows you that there are three spots inside of the ribosome where tRNA can enter and interact with the messenger RNA that's feeding through the ribosome. So those three sites are nicknamed the A, P, and E site, and that stands for the add site, the peptide site, and the exit site. And so tRNAs are going to come in here at the A site. Then at the peptide site, the amino acid that's on the tRNA is going to be added to the growing protein or the growing polypeptide. And then the tRNA will shift into the exit site and it will exit without its amino acid anymore. So ribosomes are, the real workhorse for the process of translation. So here's another cartoon of translation. This is an older cartoon, so it doesn't show us the E site, but it works pretty well for walking through this process of how polypeptides get built, so how polypeptides are synthesized. And there are three components to the process of polypeptide synthesis. This might sound a little bit familiar from the three components to the process for mRNA transcription as well. And the names are the same. So initiation, elongation, and termination. It's just some of the details that differ between these processes. So for prokaryotes like bacteria, the process of initiation begins when the small ribosomal subunit binds just upstream of a particular sequence in the RNA, an AUG sequence. This sequence is known as the start codon because it's actually the start for every single protein. So that's in bacteria. In eukaryotes, the small subunit will actually bind onto the 5' cap that's added to the messenger RNA, and then the small subunit will just feed along the mRNA until it reaches the start codon. And so the first tRNA that's going to bind at that start codon is a tRNA that carries methionine. And at that point, once the small subunit and tRNA are in place, then the large ribosomal subunit can also bind to the mRNA to begin the process of translation. And so once the large subunit, small subunit, and the initial tRNA are in place, that whole collective is referred to as the translation initiation complex. So there are 4 molecules here working together, right? 2 ribosomal subunits, a tRNA, and a messenger RNA forming this translation initiation complex. Okay, from here, the next step is elongation. And it's really going to help. We'll take a look at some videos that animate this whole process. I think that kind of helps to see that this is a continuous process. So in the elongation step, one of the things that happens is a process of translocation. So the ribosome feeds this messenger RNA through. It's kind of like a little ticker tape that feeds through the ribosome. And it does that one codon at a time. And so what happens is, the first thing that happens is the start tRNA at the start codon is going to be moved into the P site, right? So that's the polypeptide site. And that means that the A site, the add site, is empty. And so another tRNA can come into the A site. And if it has an anticodon that matches up with the codon on the messenger RNA, then it will stick in that A site. Then as this whole thing feeds along, so the mRNA is going to feed and shift 3 units over, this first tRNA is going to wind up in the E site, where it can then exit from the ribosome. And now the second tRNA will have shifted over, so it's in the P site, where its amino acid will be attached to the first amino acid, so that there will be a peptide bond between those amino acids, so the polypeptide chain will start to grow. And then once the second tRNA is in this P site, that means the A site's empty again for the next tRNA to come in and match up with its anticodon. So there's a recognition process at this A site for the next tRNA. And this whole process will continue. So the next thing that will happen is this mRNA will feed through again. Our tRNA number 2 is going to leave from the E site, and its amino acid is now attached. And now our third tRNA is going to shift over into the P site, and its amino acid will join this growing polypeptide chain. And so it's not shown here, but just so you're aware, this isn't a free process for a cell. It does take energy. So there's kind of an energy component that's occurring as well. You don't need to know too much about that energy component. I just think it's useful to be aware that there's an energy cost to making new proteins. Okay, so then that's the whole process of elongation. And as I promised, the terminator is back because this whole process then ends with termination as well. So here's what happens during the termination process. So if you think back to that chart that showed you which amino acids different codons coded for, RNA codons, you might have noticed in that chart that there were some places that were labeled stop. And so those codons, they're referred to as stop codons. They don't actually code for a specific amino acid. Instead, they are sequences that are recognized by another type of compound referred to as a release factor. And so when the ribosome arrives at one of those stop codons, instead of having a tRNA come in and recognize and add another polypeptide, a release factor enters instead. And so then as this ribosome moves further along or feeds this mRNA through further, then the last tRNA will be released. And with this release factor, then the ribosome is able to pop off and free itself from the mRNA, and the process of translation has ended. And we have a new free polypeptide from this translation process. And again, this isn't an energy-free process either, but we've eliminated showing you that just to simplify this process. So we're actually not quite done at the point where we have finished translation. So in some cases, as soon as translation ends, we may have the new protein of interest, but in a lot of other cases, there's still a more involved process for folding that protein into its final configuration, and there can be some additional modifications that take place after translation. So I've labeled them on this slide as the post-translational modifications. So just as a reminder, we're going from a linear sequence of amino acids here to a three-dimensional shape. So there can be both secondary and tertiary structure, right? So that secondary structure is due to hydrogen bonding by the amino acid backbone, and then the tertiary structure is determined by interactions among amino acid side chains. Proteins are generally not very short. They tend to be fairly long, so this process of folding a linear amino acid sequence into its final three-dimensional shape can be a really involved process. And so sometimes that process is aided by additional proteins in the cell called chaperonin proteins. You can think of that as being, like the word chaperone, something that, helps and facilitates a process. And this also highlights why if you heat up a protein and so you denature that protein, you break up its tertiary and secondary structure, it's often very difficult to refold that exact protein again. So if a protein gets denatured, chances are you're not going to go back to functional protein from there. And there can be a bunch of other types of post-translational modifications as well. So for example, there are molecules that are a combination of amino acids and carbohydrates. So those molecules are referred to as glycoproteins. And we've actually already seen an example of a glycoprotein when we're looking at blood types. So the proteins that are put on the outside of red blood cells that can serve as antigens are actually glycoproteins. So that's one example of a post-translational modification. In addition, the process of combining together multiple polypeptides, so establishing a protein's quaternary structure, is another type of post-translational modification. So we've seen an example of protein that has quaternary structure. The molecule hemoglobin is composed of 4 different amino acid subunits, 4 different amino acid chains. And sometimes some of the peptides that get added onto a protein can serve as a target signal. So a peptide might get added that signals that this protein should get sent to a specific cellular destination. And then in yet other cases, sometimes the final polypeptide that gets built winds up being chopped into smaller pieces, so it winds up being split up into different polypeptides. Okay, so again, we'll take a look at these animations of molecularly accurate versions of the process of transcription and the process of translation. I think they really help to reinforce how these processes work. And so the main things to know from this are the names of the molecular players and the stages to the different processes and how they work for transcription and translation. Okay, so that's it for translation. So now in our last video in this series, we will consider DNA mutations and different types of DNA mutations and the consequences of those mutations.